Hello again. So what I am showing you now is a different kind of representation. And so this is the same kind of drawing that I made in the previous introduction part of this recording. And so what I've done is I've used the paintbrush tools and I've gone in and I've painted this whole scene, let's say, and I'm going to take us through this scene and see what it can tell us about this embryo. What I want to show you at the outset is something that we can observe just from the surface, okay? We can see that this embryo has some shape to it. Interestingly, at the top, it's kind of flat. At the bottom, it's more curved. And on one side, we have a kind of appendage coming out. And that appendage has a specific name. It's called the dorsal appendage. And that's because it occupies a specific space in the embryo. And so there are two different axes along which this embryo is already patterned. And we have the dorsal to ventral axis, which is why the dorsal appendage is up at the dorsal side. And we have the anterior to posterior axis. And we can see that the dorsal appendage is at the anterior side of the embryo on the dorsal half. And that's very interesting because that tells us that when this egg was formed, it was already formed with a little bit of information stuck onto it. And that information is read out on the outside of the embryo itself. It's read in the differences between the dorsal and ventral side, and it's read out in the differences between the anterior and posterior ends. Now that's very important because we have to ask the question regarding that cephalopharyngeal skeleton. How did the cells that make that structure know what they needed to be at the beginning of embryogenesis? And so I'm going to take us onto the other side of this embryo and show you inside the event that took place that made that happen. So I'm just going to rotate this around. There we go. And let me just find the right position for this. Here we go. All right, so here we have the embryo. All right. And what I want you to notice is that there's some things happening on either end. Now, this is a very interesting stage of a development that's specific to the fly embryo, which is called the syncytial blastoderm. Okay, so syncytial meaning same cell, because all of these nuclei, these blue nuclei, they're occupying the same cytoplasmic space. It's a blastoderm because all of these cells now have arranged themselves onto a cell layer. So they're all in a layer on the periphery of the embryo, but they're all sharing the same cytoplasm. Okay? Something is going on here at the ends. We can see there's some material here on the anterior end, which actually corresponds to the anterior dorsal appendage. Let's look a little bit closer and see what's going on in that region. So just gonna bring this over here, and I'm gonna zoom us in. Okay. There we go. Now, I want to point out something very interesting here. There's 3 colors, and these are 3 different layers that are part of this egg. The outermost layer is the chorion, which is the eggshell. Okay. The inner layer is called the vitellin membrane, and this is a membrane made up of a protein called vitellin. It's hydrophobic, and it serves as a kind of liquid barrier to the outside world. So the embryo itself does not dry out. It allows oxygen through the dorsal appendage, and it keeps in liquids. What it also has is something that was deposited by the mother, and that something is actually information. It's information that tells the cells at this periphery what they ought to be when they grow up. And what's going on here? How does that information that was deposited by the mother on the outside of the embryo, how does that information get into this embryo? How is that information interpreted by the cells? And let's have a look a little bit closer and see what it takes for that to happen. So I'm going to zoom in a little bit farther. Okay. And I just want to focus on the outside of this embryo. Okay. So now I'm taking us into that paravidal space. And we can see we're surrounded by kind of these blue particles. Okay. Now these blue particles represent the information that was deposited by the mother. And how is that information received by the embryo? Well, if you can notice, there are these green protrusions coming out of the embryo surface, right? And each one of those green protrusions has a little bit of a blue spark on it. So each one of these green protrusions is a special protein which sits at the surface of the membrane. And that protein specifically acts as a receptor for the ligand that is out here. And so that ligand binds that receptor. And that receptor receives the information and passes the information through into the embryo. And so what I'm showing you here is something very interesting. So we are in an area where there's a lot of this outside surface signal, which is called a ligand. There's lots of ligand. And where we have lots of ligand, all of the receptors are bound to it. And you can see here this is an individual, but the ones that are bound are actually pairs. So what happens is as the receptor binds to the ligand, the two individuals that would be floating around in the membrane come together and form what are called dimers. So that's what happens when you have a lot of signal around. Let's take us back out to the outside view and zoom out a little bit, because what I want to show you here is that you don't have the same amount of that signal all over the embryo. You have a lot of it at the tip here. If I move around and come this way, we have a little bit less here. And if I come out here, oops, if I come out here, we have less still. And then if I come out to the here, we have zero, none at all. And what's interesting is that something is corresponding inside of the embryo to that outside signal. Where we have a lot of outside signal, we have a lot of this red, sparkly action happening, right? So what does that mean? What is going on there? All right, well, let me show you. Let's go back to our high concentration of outside ligand to a lot of dimerized receptor. What happens on the inside of the cell when those receptors dimerize? Well, let's come into the inside. And what we can see here is that we have some complexes forming on the membrane. So what can I show you? This is what I'm choosing to represent as a protein, okay? And I'm going to call this protein, let's make it a little bigger so you can see it. Here we go. Here we go. I'm going to call this protein RAF. Okay? Just know that that's what that protein is called. What happens is when the receptors bind to the outside ligand, there's a cascade of activation that takes place. This receptor is activated. It recruits proteins from the cytoplasm, such as these. They come in, they complex to the receptor, and they bring in the protein RAF. Okay? That's RAF. It's brought to the membrane where the receptor is activated. And what happens when that receptor is activated and all of those proteins are brought together is that the information is passed along from the receptors through these proteins now, which talk to each other. And then finally, it comes to another protein which looks like this. And this protein, This protein is called MEC. And MEC is what I have chosen to represent as all of these sparkling red proteins that are accumulating inside the cytoplasm. Okay, so MEC comes to RAF activates it, and where you have a lot of, where you have a lot of active receptor, now you have a lot of active MEC, and you can see all of that now around me. Okay, so what happens then when you have a lot of active MEC? Well, ERK. MEC phosphorylates another protein called ERK. And ERK finds its way into the nucleus, where it can interact with other proteins. And so I'm choosing to represent that here, okay? And where we have a lot of active receptor, we have a lot of this protein now that's now in the nucleus, okay? And it's able to interact with another protein, which is called Capicua. And Capicua likes to sit on DNA, and it likes to occupy DNA and keep other proteins from binding to these places. Now, interestingly, ERK can come in to the nucleus, and when there's a lot more ERK in the nucleus than there is Capicua, ERK will bind to Capicua, it'll modify it, and then it will, kick it off of the nucleus, and you can see that happening here. And then once it's modified, ERK will let it go, and it'll get kicked out of the embryo, kicked out of the nucleus itself, and you can see it kind of accumulating in this space, right? And once that happens, once the Capicua is lost from the DNA, like so, now this DNA is accessible to another protein which can come in and can activate the genes necessary to make this cell have the right identity to make those head skeletal parts. Now, it's very interesting that we have this specific cell in this specific place of the embryo. Let me zoom back out. Okay, and it'll remind us that we are in an area of the embryo which has a lot of this external ligand, which activates a lot of receptor, activates a lot of the pathway, activates ERK, and a lot of that ERK can come into the nucleus and kick Capicua out of that position of the DNA. What happens if I move over to a position now where we have even a little bit less extracellular ligand? Okay, let me just zoom down into the membrane of the embryo here. Okay, and we can see here, out here, there's a little bit less of the extracellular ligand and less of the receptor is bound to that ligand, and we have less dimers formed. If we move down now in through the membrane and into the embryo, we can see that because of that, there are less complexes formed. And then when we come into the nucleus itself, we can see that there's less ERK here now in the nucleus interacting with Capicua. And so you can have more Capicua bound to the DNA, keeping more of these sites of DNA closed off for activation. So now, as opposed to the cell that was over there, which in a space with a lot more ligand and a lot more activation and had the DNA open, here we have the DNA a little bit more closed. And so only one site, let's say, is accessible to an activator. And this cell might actually have a different fate than the cell that was over there. So what happens if I come a little bit farther out now and I take us to a port of the embryo where there is no external ligand? And we can see here, no external ligand floating around the paravital in space, nothing bound to the receptors. And so because there's nothing bound, they're all monomeric. And so they've not dimerized. No activation has occurred. That chain of events has not been passed along. And so there's no activated molecules. And then when we come into the nucleus, what do we find? We find that the entire nucleus is full of this other protein, Capicua. It's completely covered the DNA. And now this cell cannot activate any of the genes that those cells on that side of the embryo were activating. And so what is happening here, if I zoom out again, is we have seen a gradient of information, which provides through a gradient of activation a different pattern of cell identities by affecting the availability of that strand of DNA to activating proteins. Here, that DNA is completely open, and so all of the genes necessary for making the head skeleton are going to be activated in that cell. As we go away from the source of the information, less and less of that DNA is going to be accessible to activators. And so different gene sets in each one of these cells are going to be turned on. And so each one of these cells is going to have a different identity. And then here we have a cell that's going to have a completely different identity, which is going to be completely different and apart from the head. Here we have different parts of the head. Here we have one that's not even going to be a part of the head. So this is a demonstration of a phenomenon called patterning. And patterning is very important because it tells different cells in the embryo to have different identities. And that's what tells the head to develop, or the head should develop. And that's what tells the tail to be where the tail should be. And parts that shouldn't be making head structures should make trunk structures. So I hope this has served as a demonstration of two different scenarios that can be demonstrated through virtual reality, mixed reality. Obviously, my artistic skills are at a limit here. Different tools will hopefully be developed to allow for different assets to be brought into something like this. But this is just a kind of a quick proof of concept of what I would like to demonstrate here. And I hope you found it informative and at least entertaining. Thank you.